The overall goal of this experiment is to perform multicolor immunohistochemical visualization and enumeration of various T lymphocyte subpopulations in archival formalin-fixed paraffin-embedded tissue sections from oropharyngeal squamous cell carcinoma samples.
This method can help answer key questions in the immuno-oncology field, such as: is there a link between tumor tissue infiltrating immune cell subpopulations and patient prognosis?
With this technique, you can visualize several biomarkers in one tissue section.
The implications of this technique extend towards cancer therapy, as the simultaneous evaluation of multiple immune cells or immune-related factors may help in patient stratification for specific immune therapies.
Begin by using a microtome to obtain 4 micrometer thick formalin-fixed paraffin-embedded tissue sections, laying the tissue ribbons on the surface of a 45°C deionized water bath as they are acquired. To capture the sections, place a glass slide in the water directly underneath the tissue sample and carefully move the slide up at an angle. This section will stick to the glass surface as the slide is lifted out of the water. After drying the samples overnight at 37°C, deparaffinize the tissues with 3 5-minute total immersions in 100% xylol. When the sections have been completely deparaffinized, rehydrate the tissue samples with 3 descending ethanol immersions, followed by a 5-minute wash in deionized water.